It is my immense pleasure to welcome you all the faculty members from respective institutes, universities, all the academicians, and my beloved students. I welcome you all on the behalf of the Department of Chemistry, Adamas University, to this webinar entitled "The Kinetic Process Modeling of Kinetic Approach to Model Biological Processes." It is our immense pleasure to welcome the speaker of the seminar, Professor Shuman Kumar Boni, who is an eminent scientist from the Bose Institute, Kolkata. It is our privilege to hear from him. Sir, I welcome you. Now, I would like to welcome Professor Shonjit Ghosh, who is one of the most eminent scientists in India, who is a former professor in the Department of Chemistry, Presidency College, and also the principal over there. I would like to ask, I'd like to kindly say something, sir, about the seminar, and please say a few words to our student.
Am I audible?
Yes, sir.
Okay. Welcome, Suman. Are you there?
হ্যাঁ স্যার আছি
কেমন আছো?
ভালো আছি স্যার
আচ্ছা. Thank you very much for accepting.
Oh, it's okay, sir. It's my privilege.
Yeah, accepting invitation of Adamas University Department of Chemistry. I welcome you all, and it's a very pleasant moment for us that Suman has agreed to give a lecture on a very important topic these days. You know, I want to remind our students that research in these days are going on two directions. One is material science, another is life science. And people from chemistry, physics, they're all biology, all they're working together. If you want to work in material science, you may have to, go to the biological processes. And if you want the life science, then also you need the background of chemistry very much, as well as physics and computer science. Particularly in this COVID period, all we have seen that molecular biologists, virologists, biochemists, chemist people, pharmaceutical people, computer people, they're all working together. Even the AI, that is artificial intelligence and machine learning, are coming in the big way to solve these problems, particularly the designing the drug. And we are very proud that in country like India, I think 180 crore vaccines have been already given to our, countrymen. That's a very good progress in this direction. And I hope that people are working all over the world to find a particular solution, like designing a particular drug or design more efficient, vaccine, so that we can prevent this particular COVID-19. And students, you must know that experimental research is there, but theoretical research is very important in these days. Particularly if you, are not able to carry out or determine the structure of a molecule, then theoretical chemists, they can solve the problem in some way. For example, this SARS-CoV virus, their interaction with other molecules, interaction drug molecules, have been carried out by theoretical chemists and people in the bioinformatics. So quantum chemistry or quantum mechanics is coming in big ways. Along with that, in these days, AI and ML are also helping us to find the right direction. So I think today's lecture will highlight, the biological processes, the kinetic approach, because in all the processes, if you want to find the structure, that is fine. But when a molecule interacts with other molecule, then you have to know the dynamics of the process very much. Otherwise, you cannot say the functionality of these molecules or the behavior of these molecules or how do they react with other molecules. So I'm sure that Suman's lecture today will be very beneficial to our students and also to our faculty members. And I'm sure that if anyone wants to, some kind of collaboration, either a summer project or any other project, particularly the faculty members, Sumon will be very helpful. I know Sumon for a long time, and I'm sure that he is going to help us a lot. And Sumon, you are in a Bose Institute. That's a building and little garden. But if you come over to this particular campus, we have more than 100 acres, 130 acres of lands and green beauties full of flowers and trees, and all buildings are, central air conditioned. So good days hopefully are coming, and I like to have you, or everybody in the department will like to have you in physically in our campus so that we can interact with in a better way. So with these few words, Siddhita, I once again welcome Suman, and over to you.
Thank you, sir, for such enriching speech. I hope that students will get motivated from their word and also more eagerly waiting for this talk. Now, I would like to welcome our HOD of Department of Chemistry, Professor Rupam Shen. He is a very in active researcher also, and he has a very bright prospectus career over his from his PhD days, and also he was a JSPS fellow. I would like to ask him to please say a few words towards our participants. Sir.
Well, thank you, Sita. Please put yourself on mute. Okay. Very good afternoon to our speaker, Professor Suman Kumar Banik sir, and Dean sir is not there today. So, respected Professor Sanjeev Ghosh sir and faculty colleagues and students, and again, warm welcome to all of you in today's webinar, Kinetic Approach to Model Biological Processes. Today, Professor Banik sir will explore a prime important area of biomodeling chemistry. We all know there are millions of biological processes going on, all the moments in the living systems, and the processes are quite difficult to understand. To comprehend the mechanistic pathways, modeling the kinetics of the process is very important to improve our understanding. Hopefully from today's lecture, we'll learn a lot regarding the modeling of the biological processes. Before going to the presentation session, I'd like to introduce our department very shortly. Our department is very rich in terms of faculty quality. Most of the faculties are from IITs and Supreme Institute of India, and good research career, instrument facilities, and updated curriculum. And moreover, we are very lucky to get Professor Sanjeev Ghosh sir, who always help us to grow all round. So our department, actually, we have 3 branches of sciences: forensic science, immunology science, and chemical sciences. We have right now 12 regular faculty members in our department, and currently we are running 3 major district projects. And we have many sophisticated instruments here: AAS, GC, UVVs, perimeter, TCSPC, and many more. So if anyone wants to collaborate with us, we'll be happy to provide any kind of assistance. So this is all about our department, and thank you all. Now over to Sita. Sita, please.
Thank you, sir. It was a very good introduction, and it was a very good description for our institute and our university or department. Thank you so much. Now I would like to call Dr. Shudeshna Kaur, who will for onwards moderate the session to today's session. She is an assistant professor in the Department of Chemistry at Adamas University. Now I would like to call her. Please, Shudeshna Di, it's over to you.
Thank you, Srijita, for giving me this privilege. Good afternoon, everyone. A very warm greeting to all of you. In today's webinar on kinetic approach to model biological process, it's a pleasure to welcome an eminent scientist, Professor Suman Kumar Banik. He is a professor in the Department of Chemistry of Bose Institute in Kolkata. I came to know from Srijita that Professor Banik is very much unwilling to discuss about his academic journey. I beg your pardon, sir. Please allow me to share your academic journey with all the attendees. Professor Suman Kumar Banik has secured both of his bachelor and master degree from Calcutta University. He completed his PhD from Indian Association of Cultivation of Science in 2001. In 2002, he moved to Germany for pursuing his postdoctoral work in Max Planck Institute of Physics of Complex Systems. He worked there till 2004. Next, he moved to Sweden for another postdoctoral work in Nordic Institute for Theoretical Physics. After doing research work in cultivation of science for 6 months in 2005, he again moved to Virginia for doing research in Department of Physics, followed by Department of Biological Sciences in the Institute of Virginia Technology. He worked there for 2 years. There he focused on stochastic approach to DNA breeding dynamics, sequence sensitivity of breeding dynamics in heteropolymer DNA, master equation approach to DNA breeding and breeding dynamics in heteropolymer DNA. In the month of November 2008, Dr. Banik joined Bose Institute as an assistant professor in the Department nonlinear dynamics and theoretical biology. He has lots of publication in international journals. Sir, all of us have to admire your academic journey. Hope your talk will inspire all the students a lot to proceed in their academic and professional life. So taking permission from Professor Sanjeev Bose sir and HOD sir, may I request Professor Suman Kumar Banik to start his presentation? So would you please start your presentation?
Yes, I'm a bit sloppy in this technology thing.
Okay, sir, no problem, sir.
I'm
Take your
trying
time,
to.
sir. Yes, sir.
Is it coming on?
Yes, sir. Yes, sir.
Can you see the screen sharing? Wait. So share screen and the entire screen.
Yes.
And can you see the screen?
Yes, sir. Yes, sir. Yes, sir.
Can you see the slides?
Yes, sir. It's visible.
Okay, so I am audible.
Yes, sir.
Okay, so start now. Well, thank you for the invitation, first of all, and the introduction, and my regards to Professor Sanjeev Ghosh. So I'm lucky. So one of the lucky students who got in touch with Professor Ghosh during our master's curriculum and learned a lot. I mean, it was a long way. So the title of the talk is Kinetic Approach to Model Biological Processes. I would like to request the students, feel free to stop me if you do not understand anything, especially the students. And I will try to go slow. And I'm trying to develop, I will try to develop the model building, show how we work, how we use basic concepts of elementary chemical kinetics and develop models. Okay. If you have any problem in visualizing the slides, please let me know. So I'm moving to the next slide. It's basically chemical kinetics. And so we have our system of interest. This is a X. X, it could be anything. In our case, it's a chemical molecule. A molecule, it could be atom. So since we'll be dealing with biological systems, we'll be considering this X as a molecule. And so we see X is being synthesized from some source, and at the same time, X is getting degraded. So these kinetics, both the kinetics are different. So if I consider only the synthesis part, we have the rate equation. So dx dt, the rate of change of x is proportional to some synthesis rate constant. And the degradation, of course, is proportional to the x itself with a negative mu x. So mu x is the degradation rate constant. Now, if we couple these two processes together, synthesis and degradation, we have a rate of change of x with time, which is kx minus mu x and x. So for pure synthesis dynamics, we have a linear solution. These equations are exactly solvable. And for the degradation, we have a decaying function, exponentially decaying function, the solution. But when we go for the coupled system, synthesis and degradation, we have a solution which is neither linear nor exponential degradation, but it shows a kinetics which is hyperbolic in nature. So next we move to protein DNA interaction. So nowadays, I mean, all the school books have these description, biological textbooks have the description of proteins and DNA. So DNA is basically a double helix structure, three-dimensional double helix structure. So I'm just considering a 2D projection. So these are two intertwined helices of DNA. And it is said that nowadays everybody talks about the gene and DNA and disease and some phenotype, genotype, all these words are just floating around us, especially in this time of COVID. So what is a gene? The gene is basically a part of DNA which encodes some property. So for human beings, say this portion, this box portion encodes basically the color of the hair. Okay. And if I take some other part, it might encode some other property. So I say the skin color. Okay. So now this structure is heavily complicated. It has a lot of interaction. So we will just take a very simple approach to describe the DNA. And so what is that? So we'll consider this is the gene of our interest, and it has some upstream part on the negative on the left hand side, and on the right hand side we have a downstream part. So the upstream part basically is the place where several molecules, it could be these are protein molecules, of course, it could be RNA polymerase or some transcription factors. So the transcription factors are biomolecules which starts the process of transcription. And this side, this point, this arrow-headed part is called the transcription TSP or transcription start point. So what happens is basically a big blob of protein, the DNA polymer, RNA polymerase comes and sits over here and it reads the frame. So they read this frame, and which encodes the information of a certain characteristic in the form of base pairs ATGC. So given this background, I will now consider a situation where this gene is off. Okay, so the gene is off, and now I will bring in a molecule which I will consider as a transcription factor. See this purple color circle? Okay, so these are basically cartoons. This is not exact process. So these are cartoons to just give a simplified version of what is going on inside the cell. And then this guy, this purple ball, will try to attach itself to this upstream portion. And so it's a reversible process, and we have an on state. So when it is on, when it is bound to the upstream state, it will bring in RNA polymerase, which I have not shown for the sake of simplicity. And the gene will be, the RNA polymerase will come and will read the frame, will produce the gene product. Now, can we model this process of on and off state transition? So this double-headed arrow means this process very fast process. It happens in a time scale of few seconds. Now, if I have the probability of off state as pi inactive and the probability of the on state as pa of the gene of interest, and x, I have shown earlier, this x, so the inactive state incorporates the transcription factor.
Sir, sir, it was actually, we cannot hear anything for the last 1 or 2 minutes. Sir, am I audible, sir?
Hello, sir. Maybe there is some problem in his side in internet. Sir, can you hear us? Maybe any network issue from his side. Let's wait a little bit. Maybe there is some network issue. Sir will join after a little bit time. Please wait.
ওকে, আমি শোনা যাচ্ছে এবার?
স্যার এবার শোনা যাচ্ছে।
আমাদের নেটওয়ার্কটাই ঠিক এই সময় আমি এই ভয়টাই পাচ্ছিলাম।
হ্যাঁ স্যার, কোনো
আমাদের
ব্যাপার না।
ইনস্টিটিউটের নেটওয়ার্ক ঠিক এই সময়টাতে একটা সার্জ হয় এবং পুরোটা বসে যায়।
ওকে স্যার, নো প্রবলেম।
সো কতটা অবধি শোনা গেছে? আগের স্লাইডটা কি পুরো বোঝা গিয়েছিল?
না স্যার, এটা একটু এক্সপ্লেন করলে ভালো হয়, এটা পুরোটা শোনা যায়নি।
কতটা অবধি বলেছিলাম আমি?
স্যার অফ অন স্টেট, কিন্তু প্রবাবিলিটি ম্যাথমেটিক্যাল এক্সপ্রেশনস গুলো একটুখানি আর কি।
ওকে, সো coming back again, so we had this off state of the gene of interest and the on state, and so if we define the on state probability, off state probability as pi, and the off state probability on state probability as pa, so inactive and active state, and x as a transcription factor, this purple blob. And so we use two conservation equation, pi plus pa equals to p. Then we can write the time evolution of this probability states pi and pa, where the kb is the binding constant of binding of this transcription factor to the promoter site or the upstream site, as I have shown here, and ku is the unbinding rate constant. And if we solve these two equations using this conservation relation, we will arrive at, it's very simple, very straightforward. We will arrive at the final expression of p active and p inactive, where these small k's are replaced by the capital K, which is known as the binding constant in the typical biological textbooks, standard biochemistry books. We will have where K is basically the ratio of these binding, unbinding, and binding constants. So given this background, so we now know the time evolution of X, and we also know the probability of the system being in the on state and in the off state. So we are ready to talk about the gene expression. Okay, I'm back again, sorry. And so the gene expression, so we have this cartoon again. We have the signal here. So the signal is the purple blob we have shown in the previous slide, which is basically a transcription factor, a biomolecule. And this is our gene of interest. The signal will come in and we will have lot of gene product after the transcription and translation. And so we can write the rate equations of the signal ds dt equals to ks minus mu ss, which is now, I mean, for the students itself, don't get confused with these s and x. I will keep on changing them. They are just dummy variable. Okay, so now we have this ds dt. It's ks minus mu ss, which is basically the synthesis and degradation of my signaling molecules, and x is my gene product, these black circles. And so the black circles will be produced only from the on state of the gene. So I have used s by k plus s, where I have actually in the previous slide I have shown the probability of the on state, which was x by k plus s, just replaced x by s because now my transcription factor I have designated as s, and it's the normal degradation of my gene product. Now the question is, in a multicellular system or in an isogenic population of single cell collection, do all cells behave in the same way? So the answer is no. So it's a very nice picture from the experiments from Eloyd's group, probably at Caltech. So they have expressed some reporter GFP in single cell bacteria. So this is a population of bacterial cells where this GFP has been expressed, and you can see some of the cells are glowing nicely, but some are not. Some are showing mid intensity. So we see a variability. We see a variability in a genetically identical population. So genetically identical population means we have a collection of bacteria. It could be all the bacteria could be an E. coli cell. Now we know this name. It could be some other bacteria. And the reaction kinetics must be probabilistic in nature, because if the reaction kinetics are deterministic in nature, then all the cells will grow with same intensity. But now we see in this experiment result, we see that the intensity is not that great for some of the cells. So it must be something going on inside the cell, which is showing, or rather indicating, that this reaction kinetics that I have shown here, written here, the transcription factors are coming in, getting produced, and they're coming in and binding to the gene of interest and producing our gene product is probabilistic in nature. So let us see what is happening. So if one carries out some simple simulation of those reactions I have shown in the previous slide, which are based using some Monte Carlo simulation, popularly in a variant of Monte Carlo simulation known as stochastic simulation algorithm, or popularly known as Gillespie algorithm, we see that all the cells are not behaving in the same way. So this is the average value of the number of protein level. So it's basically 100, say for simplicity. Sorry. And then we see it's dancing around this mean value. And if we take like lots of cells, like 900 cells in our system, we will see that most of the cells are not glowing very high. So very few cells among 900 cells are around that peak, n equals to 100 value. Some cells are not expressing the gene product at all on the two far ends, on the left hand and on the right hand. So that means the cells are behaving or expressing their gene product in a noisy manner. So this is a nice cartoon. It's a nice article, popular article published in Nature in 2008, which shows this a crying baby inside the cell. So how do we do that? How do we explain? So going back to a very standard model of random walk. So this is a very standard and one of the finest books written on random walks concept in biology by H.C. Berg. So Howard C. Berg is an experimentalist. He worked like for throughout his life. Excuse me, just stopping for a moment. Yes. আমি একটা সেমিনারে আছি, একটু পরে ফোন করছি আমি। And so he worked on bacterial motion, and so given all the information in that book, I'm just using a very simple model of random walk in a one-dimensional lattice where we have this n state, n+1 state, n-1 state. What this state means that a particle is moving, hopping around in this one-dimensional lattice from 1n to n+1, or from n-1 to n. And if we just express this, I will just say it for the purpose of developing the model, but it's a bit complicated for the students, I understand. And so we have this master equation. Now these P's are the probability, and these are probability states. This dot over the P means time derivative, as we said, rate of change of Pn. So Pn means n is probability of finding the system in the n state. Npn minus 1 is the probability of finding the system in n minus 1 state. So this shows dynamics or kinetics. I would say use the word kinetics rather than dynamics. So the kinetics is a discrete kinetics. It shows the discrete kinetics. So now if we write this master equation in the continuum domain, we have the Langevin equation. Now you see, except this xi nt term, we have this n dot equals to g minus rn. So we have seen this equation before in the previous slide. So replace n by x, you will see this dn dt or dx dt equals to g, means some source is giving producing the n and it is getting degraded. Now this xi n, why this xi n is coming in? This xi n is coming in because of this random walk nature. So this the particle is executing a random walk in the system, or it's diffusing inside the cell in a completely random manner. So to consider this randomness, we have brought in this function xi n. So n is the index for the n, it could be anything. So xi, since it's a random process, it should have some statistical properties. Any random process must have some statistical properties for its complete description. So the average of the xi n, is 0, so it has 0 mean, and the variance, this is the 2-time correlation. If we put t equals to t prime, it would give us some xi squared t. The variance is basically g plus rn. So growth term plus the loss term, and this is Dirac delta function. So this is a typical expression for noise processes, and because of this particular nature of the noise processes, it is basically Gaussian noise. So the Gaussian noises are the simplest noise processes one can handle. There are lots of other processes which is not important right now. And then we will bring in another thing. Since we are dealing with random walk and random processes, we need some other metric or measures, which is entropy and information. We all know, I mean, in our undergraduate class, and nowadays probably in the +2 level, students learn about entropy in thermodynamics. So I will just use the outcome of repeated experiment. So what could be a repeated experiment? The coin tossing would be a repeated experiment, or measurement of the gene product in a experiment. And like if you have it's like thousands of gene expression, as shown in the Elowitz's experimental figure, we have seen that lots of cells are showing high intensity, some are showing mid intensity, some are showing very less intensity. If we measure this intensity of the cells, we will have something like we have performed the experiments repeatedly on a single system. Since they are isogenic system, so we have an expression. So this is an expression of entropy used by like Claude-Shannon in the context of telecommunication problem. So since it's a random process, and random process, whenever we have some random process, we ascribe a distribution probability function to these processes. So our system of interest is X. If our system of interest is X, X could be intensity, of the reporter gene. So we will have a px minus px log 2 px. This is a definition of entropy introduced by Claude-Shannon. And if we have two different variables, say we have in the cell, we have expressed GFP, green fluorescence protein, and CFP, cyan fluorescence protein, we can write the joint probability distribution. So given these expressions of entropy and information, we can see that in a cell, we have some signal coming in from outside, and some noisy thing is going on inside the cell, and then we have an output. What is that output? Output is that expression of GFP level. So it is basically, it looks like the signal, the external signal, the cell is processing its information and giving us some output. So since this process is going through a noisy channel, okay, so we need a probabilistic description. And we bring in, or rather following Shannon, we bring in another measure called mutual information, IXY. So what is that mutual information? So we go back a little bit of basic mathematics again, and we have this purple circle which represents my entropy of X, and the green circle, the entropy of Y, and together we have this joint entropy of X and Y, and this overlapped region is the mutual information. So what exactly this mutual information is? Now, going back to this definition of Shannon's definition of entropy, we can say that Shannon's definition of entropy actually gives us the uncertainty information, uncertainty in measurement. So we are, even if we carry out the experiment several times, we will never get the exact value. We will always have some uncertainty associated with it. So now if we have this Hx and Hy, so Shannon mutual information basically tells us, given the knowledge of one entropy state, what is our chance to predict the nature of entropy of the other variable Y. So now this is the mathematical expression. So basically we have just used these entropy definitions here to write this. This is very straightforward. And now in the previous slide, I have shown that our noise process is Gaussian. So we had this average property and this two-time correlation property, or the variance property of the noise processes. And so for Gaussian noise processes, this probability distribution function, the joint probability distribution function, or the marginal, so when we have single one, the marginal one, this can be replaced by the variance and covariance of these variables. And this is the full expression of mutual information. There's a typo, it should be semicolon here. And this ratio of the variance and covariance gives us the signal to noise ratio, or fidelity of the system. So the fidelity gives a measure how smart the system is, how smart the bacteria is, how smart the cell is, because the cell needs to be smart to process the input, because something going on inside in a noisy manner, it reads the signal, it does some processing inside and give us the output. It could be like a cell, mobile phone we use. The mobile phone captures the signal from some tower or some source, and then we have something is going on, some black box. We have this, we never open up the mobile phone inside and don't check the circuit. So something is going on there. There's an intertwined circuits and some complicated structure. It's something going on. And then we have the output, which is basically when we talk or we listen, we watch movies, we watch videos, we listen songs, all this. So signal to ratio is a very important measure to check whether my cell or my biological system is smart enough to process the extracellular signal. Now, given all these backgrounds, I will just bring in a very simple system of two-step cascade. So what is this two-step cascade? So the two-step cascade is we have three gene product. So S, X, and Y. And they are interacting with each other in different ways. So the direct arrowhead means S is activating X, X is activating Y. In the next diagram, S is activating X and X is repressing Y. Means X doesn't like Y, so X wants to repress Y. And so similar, we just took all permutation combinations and we have these four structures. We'll say the first one is activation system, and then activation repression system, and then repression activation system, and repression system. And now we need a little bit of mathematical description for them. I'm just bringing in, it must not be scary. So we have the time evolution of S, our one gene product, the time evolution of X, another gene product, and the time evolution of Y, another gene product. So I've just used very simple, the general expression for the synthesis term, and we, of course, we have the loss term. And for each of these variable S, X, and Y, we have associate noise processes. And as usual, these noise processes have zero mean, and the variance is basically this. Now we bring in this Kronecker delta. That means these noise processes are uncorrelated with each other. So the noise process of S is not affected by is not correlated with the noise process of X. Of course, it gets affected by S affects Y, sorry, S affects X and X affects Y. And so we have this simplified expression, and we have used these functions. These are the explicit form of the function of this Fs, Fx, and Fy. Now we see that since we have, it's a linear synthesis and degradation, but for X and Y we have complicated structure. We have this K function because it's getting, S is getting bound to X to activate it. In some processes, S is bound to X to deactivate it. And these N guys come in because of Hill coefficient. And those who have studied a little bit of biochemistry, I think nowadays they teach it in undergrad level in common chemistry course. So basically, N is the Hill coefficient, which represents the cooperativity. So if N equals to 1, we have single molecule, one S bound is bound to X promoter region, and N equals to 2 means two molecules of S, either in the form of monomer or in the form of dimer, is bound to X promoter. So we have these functions, and the next slide a bit very technical. So we do some, so we see that if we have these N equals to 1, these are nonlinear functions. These are not exactly solvable most of the time. So we need to linearize them, and for this linearization we use a very technical aspect, which is linear noise approximation. So what we do is we have the steady state expression of each variable, and each variable steady state expression average population at steady state. So Z is some random variable. We make an expansion around the steady state and do a little bit of math, very standard math, and we arrive at some matrix which is known as Jacobian matrix, and from this Jacobian matrix we can write equation called Lyapunov equation. So this is very technical, I must admit. So given all this technicality, we have, if we now solve this Lyapunov equation, in our case, since we have linearized around the steady state, it is exactly solvable. I'm not going to write the expressions of variance and covariance because it's more scary, and just showing you the result. And the result is this. And so what is happening here, we have some net synergy measure. Net synergy is basically, since we have these three variables, one can write mutual information for three variables and mutual information for these two variables taking combinations. So this measure means delta I could be 0, greater than 0, or less than 0. When it is greater than 0, we say it is synergistic. I didn't write it here. Synergistic means X and Y are acting synergistically together for S. And if it is negative, that means either X or Y, it is redundant to interact with S. So, and we have this SNR measure for this activation and activation repression pair. I will come to that expression again. And we have the SNR expression for the repression activation and repression pair. So now let us look at the figures, our theory and numerical simulation results. So these lines are basically the theoretical calculations after solving all these moments and getting the expression of moments. And these symbols are basically the simulation results we perform using stochastic simulation algorithm. And then we see that for n equals to 1 case, that means the single one molecule of S is activating X, one molecule of X is activating Y, and so it is for the other cases, like one molecule of the variable is necessary. We see there is an overlap of results or kind of degeneracy we see for activation and activation repression dynamics. And for repression activation, we see another kind of degeneracy or overlapping of the results. And this SNR, we see the same feature. Now when we do n equals to 2, n equals to 2 means 2 molecules of S are necessary for the activation of X, and 2 molecules of X is necessary for activation of Y. And we see at low value of the signal in the x-axis, we are changing the signal copy number. So copy number means the molecules, copy number per unit volume. We see there is a breakdown of this between simulation and theory, because for n equals to 2, the nonlinearity of the system is so much that even after linearizing the system, we cannot remove that high level of nonlinearity. But as we go in the higher copy number regime, which is very close to macroscopic kinetics, we see that they actually kind of gets agreed with each other's theory and the simulation. And so what is happening here, we see the SNR for AA and AR are same, and SNR for repression activation and repression are same. So these are the, we have a very big expression of this signal to noise ratio, the fidelity, and we see that the fidelity, we did some approximation, we arrived at some simple functional form, and we see that in both cases, the activation and the activation repression are same, have same expression. On the other hand, we have the repression activation and the repression expression for the signal to noise ratio or fidelity have the same kind of expression. And we see that the activation, and both activation repression have marched at the high copy number limit. But what happens, how do we explain the difference at the low copy number limit of the signaling molecules? This region, this region, this region, and this region. So we bring in some cartoon. This cartoon says this is the first step after doing these mathematical calculations and the expressions. We looked at the functions and we have figured out the first step is the rate determining state of the whole dynamics. So now if we look at the activation and activation repression case, the first step is basically the activation. So S activates X. That means we have our system gene of interest, gene X, and S is coming in, S is getting bound here. So this is a case for n equals to 1. And then we have another site, as I have been talking about this RNA polymerase guy. This is the promoter region. They come and sit together and they start producing the so-called mRNA because of transcription, which then gets translated into X. Now for the next pair, repression activation and repression, the first step is always the repression. Now what happens actually? The repression, in the repression case, this S guy, the signaling molecule, not only sits on the upstream region, it may have, or it has a high probability of getting bound in the downstream region also. So when it is bound in the downstream region, when it is bound to the downstream region, this RNA polymerase cannot read the frame. It is blocking the road of reading the frame. So we have low level of mRNA and we have low level of X. And that's what we see here. Now we see this activation and activation repression, these two systems, these two-step cascades, although they have different kind of dynamics in the second step, the first step is the rate determining step and shows that it promotes activation, it promotes the gene expression, whereas the second pair, the repression activation and the repression system, we see the first step is the rate determining step, which is basically the repression dynamics, and we see because of this difference in the first step of the rate determining step, there's a overlapping of the activation and activation repression dynamics. And similarly, we have overlapping of repression activation and repression dynamics. And we were lucky enough to publish it in a standard journal of theoretical biology. And so I'm done here now. I would like to thank all my students and my friend from Calcutta University, Professor Pinaki Chaudhuri, and of course Bose Institute, which has funded me. And this is the nice picture. Now we have this crying baby is getting divided into two cells. One cell is crying again, so giving some expression, and this nice sleeping cell, which is not giving expression at all. And thank you. And I'm ready to take questions.
Thank you, sir. Thank you very much for this wonderful talk. Hope all of us have been enriched a lot. Now we can start our interactive session. So if any one of you have any questions, so please ask our professor. Okay, I can ask Professor Shonjit Ghosh sir first. Sir, if you have any question or suggestive information, so please discuss with sir. Sir, are you there?
Okay, can you hear me?
Yes, sir. Yes.
Yeah, it's a nice lecture and dealing with the kinetics, modeling. So, Suman, it would be nice if you give some more examples, one or two more examples.
Well, I mean, the models I have used is a typical model of gene regulatory network. So I've used those very simple ones. We are building models on some recurrent motifs of gene regulatory networks. I mean, if you look at the gene regulatory networks of E. coli or yeast, budding yeast, you will see different kind of recurrent motifs. And these motifs show very interesting dynamics. So now we have we are going a step ahead in this model. We have used some direct path, and we have this feed forward loop motif. So the idea is to understand how the information is getting processed within a gene regulatory network. That's the main idea of our work. And well, I mean, it can be done, checked experimentally. What you need is finally say we have this assay, use a reporter assay, make a reporter assay, just fusing GFP or YFP or CFP with Y. One can go for co-expression, or one can just remove the use the promoter part of the Y and fuse it with GFP and measure the intensity of GFP. And one can perform these experiments in typical setups like FACS, flow cytometry experiments, or microscope. And one can exactly measure what is going on. One can people are working on this field and in this field, and they're doing really good experiments. So I'm just running around. I'm just trying to motivate my colleagues in Bose Institute to do experiments. That's what I can do. I cannot do experiments on my own.
Just a simple question: can this be used for any imaging technique? You know, your modeling and ultimate results.
Okay, so
Using the
imaging,
GFP.
yeah. So this information theory has been widely used in neuroscience. The most in neuroscience, people have used this fMRI data or some EEG data. They have collected them, and then they have used these to figure out how the neurons on the neuronal network, how it is getting processed, because it's most important there, right? How our brain works, how our neuronal circuits or our neuronal network works. So there are experts in worldwide who does theory and experiment. They collaborate with each other, and so actually this information thing is I have learned from their work, and I tried to bring into simple gene regulatory network. In imaging, you can do that, because in imaging you have this these pixels, random pixels, right? I mean,
right.
And then these random pixels can be, you can do the statistical measures, and you can perform calculate these metrics.
Sudeshna, thank you. I think you can go to the students for questions.
Yes, sir.
Okay. Thank you for your nice lecture once again.
Welcome, sir.
Thank you, sir. Actually, today, Dr. Orkun Manna, professor in chemistry, chemistry department of Tarakeshwar College, is also present today with us. Actually, he was our former HOD. Thank you, sir, for joining. Sir, you can interact with Professor Shuman Banik if you wish.
Thank you, sir, for your nice lecture. Actually, I am also an experimentalist, so many of the things actually I could not understood. But one thing I can ask you that you have told in bacteria we know that RNA transcript can act as a messenger RNA, but in case of eukaryote, I have heard about the pre-mRNA. So in case of this eukaryote, how do you handle this model actually? Because in pre-mRNA, we know that there are exons, interions, or lysines also it has to undergo.
Oh, okay. I understand. No. These complications have been taken care of. People have already. I mean, in like I would say like almost 15 years back, people have already worked in the expression of gene expression of bacteria and eukaryotes. Of course, lower eukaryotes. Initially, they worked on lower eukaryotes like yeast, and then finally, nowadays, people are working in mammalian system. So mammalian system, now people are looking at mammalian system, they're doing measurements. So and the problem you're saying is typical biochemistry problem and it has been taken care of. So these are not a big issue.
Okay. And another thing is that you have told about the activation and then again activation. So things is that
Some noise is going on.
so in that case, actually, my question is that means while you are doing experiment, means how you can identify this process that okay, this is an activation process, and then this next process will be activation process because
Oh,
experimentally,
you just I understand. So your point is how do you know how whether it is going to be activation or repression,
yes,
right? I mean
yes.
you do synthetic biology. So you basically cut paste things together in a simple biological framework. There is a branch called synthetic biology. You do simple molecular biology and you can construct this framework. So it's a bit tricky, but it's not undoable.
Thank you, sir. Thank you very much, sir, for you as a nice lecture actually. Thank you very much. Thank you, Sudhashna.
Thank you. So Srijita has some question. So Srijita, so you can ask your question to sir.
Yes, sir. Sir, basically the thing is that as I am also a theoretician kind of, so I just want to ask about this information thing. So we can proceed, we can calculate the information for a particular cascade. We can have a control also, theoretically it can be done. So I'm just asking if there is any experimental realization or not, experimental thing there.
Oh, people have actually measured. People
Yes,
have actually measured.
it's when means control parameters are there, so they can actually control the information thing. So we can.
Yes. Exactly. They have measured. They have control parameters by some changing some like in biological some biochemical parameters. And I didn't go for it because so say for example one important parameter would be your binding constant, which is the ratio of the unbinding by binding constant. So the binding constant, if you do a change in the DNA sequence of the system of interest, you can change this binding constant, the big K. So you can actually vary this big K and measure how the system is behaving. Okay, so one control parameter could be a binding constant. Another control parameter could be the rate of synthesis. This Kx or Ks at the very beginning in the synthesis part. So you can do that in the form of at the ribosomal level. So these are all known facts. I mean, like 30 years back, it was a bit challenge. Now people know this experimental technique, and if, of course, theoretically, I mean, there is a very nice textbook on theoretical systems biology by Uri Allen. It's called Systems Biology, Introduction to Systems Biology by Uri Allen. And it tells everything, or how to control the parameters, how to tune the parameters, and How do you basically change the sequence? So these are a bit tricky. I mean, from an experimentalist point of view, it is tricky, but not undoable. They do, I mean, of course, they are leaders in the field who does these experiment quite often, and they look at the system from all these like measure the fluctuation level. So what do you need? I mean, for the mutual information thing, you don't need too much. What you need from the experiment, you need a big time series, say the gene expression, this fluorescence intensity. As Professor Ghosh was asking me, the imaging. So imaging, if you have a imaging, in the imaging technology, if you have the intensity of these pixels, if you have the time series of the pixels, so you can have this random time series of the pixels, you can bin them and you can calculate basically the probability distribution. You don't need to know whether this distribution is Gaussian or non-Gaussian. You just calculate the distribution, you can calculate the joint probability distribution, you can calculate the marginal probability distribution, and you plug them in Shannon's formula, and you can actually measure the mutual information between two different variables. So from theoretical point of view, it is very easy. From experimental point of view, it is challenging, but people are doing experiments already.
Yes, sir. Thank you, sir.
Thank you so much, sir.
Welcome. I'm happy.
It was.
I don't think it was too technical. I tried to make it very non-technical, but somehow I couldn't get rid of the technical parts.
No problem.
Okay.
So it was really an interesting session. In recent years, very few scientists are working on computational method and tools to explain complex biological processes and on their improved investigations. Deeper understanding of the concept and theory will definitely help researchers and enthusiastic students to develop new ideas and to proceed in versatile direction in individual research field. We will share a feedback form with all the participants shortly. Please fill it up and submit so that we can provide you the certificate for joining this webinar. Now I would request Dr. Srijita Talukdar to convey the vote of thanks to our invited speaker, Professor Shumon Banik. Please, Srijita.
Yes, thank you, Shudeshwari. So it is my privilege to give the vote of thanks to the speaker, who is basically he is my sir, is my respected sir. We have worked together in my very early days in PhD life. I know him. He's a great person. I know, and also definitely a very good researcher, a very good teacher indeed. I am very privileged, and I don't find, I think, the exact word to give him thanks for giving such a enjoyable session, share such an enjoyable session with us. It was a very good, very enriching lecture. I hope that students will motivate to work in theoretical biology as well as computational one, and which is, I think, students initially, I feel students have some fear in that portions, but I think they will motivate the lecture will motivate them to at least they would be curious to find something new from here. So with this word, I definitely I want to
Srijita,
thank. Yes, sir.
Pinaki is here. Can you
Yes,
ask
sir.
Pinaki to say a few words? No words to this.
Yes,
Pinaki,
sir.
Thank
please
you,
encourage our
sir.
Yes,
students.
sir. Of course, sir.
Thank you, sir.
Sir, it is very.
For me, Kumon,
Yes.
Kumon, তোর সম্বন্ধে কি বলবো? Well, I mean, for me, it is extremely difficult to say about Shumon in a formal setup. Basically, we have been colleagues since our ICS days. We did PhD at ICS together, sitting in the same room number 97.
Sir, may I interrupt you? So can you please, both of you can turn on the video. Sir, Shumon sir, can you please turn on the
You
video?
can't see me. Actually, I have some issues
Okay,
Okay, okay, okay,
okay.
okay. So it's fine, sir.
Okay, well, okay. For Shumon, since I have a long association with him since 1995. Initially, it was basically friendship, and we used to discuss our problems. Shumon worked with Professor Dev Shankar Rai, and myself worked with Professor Shankar Prasad Bhattacharya. But of course, there were overlapping areas. But things took a turn for the better from a professional point of view. After Shumon joined Bose Institute in 2008, I joined Science College in 2005. I still remember Shumon telling me about breathing dynamics. And Shumon, I think we just thought about clubbing optimization techniques into breathing dynamics so that we can get something meaningful. And we tried with a simple one-dimensional model. That was before Srijita joined. And we just tried to see if it works or not. And I still remember the first effort was published in Journal of Physics A. And of course, that was the beginning of this professional friendship. The personal friendship has been there since 1995. And since then, it has been a continuous collaboration, enjoyable. We get to meet each other every day because, because my campus is in 92 APC Road and Shumon's is 93 APC Road. So that makes it even easier. And we have explored ideas together. Of course, there are a lot of things that can be done. I mean, Shumon is the one, I should admit, that who basically pushes all of us to do things in a newer way, to think in a newer way, and 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 basically all those things, and these cascades and all those one step, two step, and this mutual information, and basically the direction of the process that we can get from kinetic measurements. These are really, I mean, in-depth study of kinetics. I mean, in-depth, thorough study of kinetics. I should admit, and at a very fundamental level, if someone wants to know about a process, this is the way it should be done. And Shumon continues to give us new ideas.
Yes.
Shumon,
Yeah,
and
I have lots of wild ideas. Yeah.
those basically wild ideas have inherent within them certain aspects which I must admit quite outstanding. Some of the aspects, and definitely worth looking into and have a long investigation to see how things turn out. I mean, maybe I don't know these things, cascades and all those things. Maybe the next step would be if we could club some kind of AI. Maybe we have a little bit of plan, neural
I think
networks, and maybe
I will try to Pinakita, I will try to figure
yes,
out. I
yes,
have seen some paper on image processing,
yes,
as
yes, yes,
Sam
yes,
was asking. I will
yes,
try to get those papers, references of articles, and I
yes,
will send it to you. I don't have the email, or maybe to Srijita, then she can actually give it to
yes.
Professor Ghosh. And
Okay,
in
okay,
addition,
okay.
I would like to say something, sir. We have a very small MSc PhD program in Bose Institute. So we have four specialization in the life science. So one is plant science, naively speaking. One is in molecular biology and cell biology. Another one is biophysics, and the final one is computational and systems biology. So we do get great students sometime, and they do nice experiments. So actually, one of our students now, he's doing postdoc. I keep on, as I said, I keep on bugging my colleagues here, and like several years back, I think it was like 10 years back, a paper came out in PNAS which shows that these some input-output relation in some bacterial communication system. They have shown this input output system is very robust. So given if we change the input from outside, the system adjusted itself in such a way that the output remains the same. It doesn't get disturbed. So I convinced one of my colleagues, he's a great guy, Professor Jayanto Mukhopadhyay, of course, in the chemistry department. And he did a really nice experiment. He used these recombinant techniques in bacteria, and he actually showed some dynamics, some experiment results using expressing some signaling system of Mycobacterium tuberculosis or TB in E. coli cell by using recombinant techniques. And yeah, I mean, so it can happen actually. It's not that difficult. In India, we always complain we need a lot of things. We actually don't need a lot of things. I mean, of course, I mean, Professor Ghosh has done excellent experiments in a very in limited setup. So, I mean, even in cultivation, we have seen how people works. So, yeah, so it is possible, sir. Okay, Srijita, thanks a lot. I really enjoyed it.
Thank you, sir. Sir, could you then kindly share that how our students can actually means go through there is an I think admission test over there in the your institute.
Oh,
So
nowadays they have changed the policy. So few years back we used to incorporate students from undergrad level so we used to take these exams, entrance exam. Now we are taking students from, there's a program called JGBILS, which is as like JAM or JEST. JGBILS is conducted by NCBS. So if you have cleared JGBILS, you can, when the ad comes out, you can always apply. But one word of caution is in the biological program, there are lots and lots of biology. So the chemistry or the non-biology guys, they get confused. Sometimes they perform really poor. And so it's not their fault. I do understand, but we keep on encouraging them. Sometimes we keep on bugging them. I never encourage anybody. So no, it's tough. It's tough for them unless they are from biological science background, because there is so much phenomenology in biological science. The students they get easily confused and lost. So yeah, it happens. Especially if a student comes from physical science background, like physics, chemistry, engineering science. So they really face a tough time here. So now, I mean, during MS course, they used to get some fellowship, but last 2 years we couldn't take any students because of the COVID, and we had problem, like it's a logistic problem of class and practical. So probably we will start this year again sometime in May, June, so the students can always apply. And we do have also a program in physics, MSc, PhD, and it's also very good. The students can always apply. They can appear for the interview, and we will see. I mean, we got really good students from non-biological science background over the years.
Okay, sir, thank you so much, sir, for this very valuable information. Sir, Professor Sanjeev Bose, sir, would you have to share any other words with the speaker?
Suman, Suman, it's a very nice lecture once again. And
thank you, sir.
Hope to see you in our campus sometime.
আমরা তো শিফট করে গেছি অলরেডি সল্ট লেকে।
হ্যাঁ, ওখান থেকে কাছেই।
হ্যাঁ, ওখান থেকে কাছে, বাট আমি এখনো শিফট করিনি, আমি এখনো রাজাবাজারেই আছি। I like Rajabajar a lot.
আচ্ছা।
It is much more green inside. I think, I mean, you have come here, right? I mean, you have visited our Rajabajar campus.
Yeah, Rajabajar campus I know, but I have also seen your campus in
Salt Lake campus. Yeah, that's the unified academic campus.
Salt Lake I have not seen. I've seen the Kankurgachi complex where Babluda and Siddhartha.
Yes, I know. Yeah.
Pinaki is our one of the member of the board of studies and helping us a lot designing syllabus
Okay.
and things like that. So one of my best students, Pinaki. So
Yes.
he's helping us a lot.
Pinakida was also best student during his PhD days.
Yes, the best teacher also. One of the best
Yes,
teachers.
he's a great teacher too.
Okay, Suman, see you sometime.
Okay, thank you,
Okay.
sir.
Okay, sir.
Okay.
Thank you. Thank you. Thank you.
So I'm
Thank you.
just going out, okay? Thank
Okay.
you.
Thank you. Thank
So
you,
thank
Srijita.
you.
Thank you.
Yes, sir.
Thank you.
Thank you. Thank
Thank
you
you. Thank you.
all. And thank you
Thank
for
you.
all the participants for joining. So
Thank you.
this is the end of the session. Hope to see you again in some other another platform with some another lecture here. Thank you. Thank you for joining.